She is currently a senior scientific advisor and principal consultant. She supervises a team of scientists at SimCip who extrapolate in vitro data to predict in vivo pharmacokinetics in humans. She also helped develop and implement models within the SimCip simulator, which links the processes of drug discovery and development using simulations in virtual patient populations. She's also involved in consultancy projects relating to the application of SimCip simulator within the pharmaceutical industry. Dr. Roland Yeo received her bachelor's in physics from the University of Natal, completed a PhD in drug metabolism, a postdoctoral fellowship in childhood leukemia, followed by a year fellowship in clinical pharmacology and therapeutics at the University of Sheffield. She has also published more than 50 peer-reviewed journal articles. Karen, welcome to today's event. I'll now turn it over to her to begin today's presentation.
Okay, I'd like to thank you very much for that very kind introduction, Suzanne, and welcome you all to this webinar. I'm just going to take you through the content of the session. Here, the first thing I'm going to start on is actually looking at the background to mechanistic PBPK models, to give you some background to that before we actually go through some of the examples. The first example that I'm actually going to cover is one involving repaglinide that is metabolized by 2C8, 3A4 and also has transporters. And therefore, what we're going to be looking at there is predicting complex DDIs involving CYP enzymes and transporters. In the second example, what I'm going to be doing is actually covering a real-life example just to basically demonstrate the top-down bottom-up approach and show you how we can actually apply PBPK modeling in the drug development process. And then finally, what I'm going to touch upon, just really a few slides, is a regulatory perspective with the regulatory perspective with respect to PBPK modeling. So if we, first of all, focus on the background to PBPK modeling, I'm also going to cover aspects such as the incorporation of variability. And if we have a look at this first picture here, what we've got is on the left-hand side, we've got things such as intrinsic and extrinsic factors. And on this side, we've got the PBPK model components. Now, this was actually a publication by Peng Zhao at the FDA that was published several years ago. But what they're trying to show you here is that if you have a look on the right-hand side with the PBPK model components, within that PBPK model, what you've got is the system components and you've got the drug-dependent components that include metabolism, transport, passive diffusion, protein binding, and so on and so forth. But what we're trying to show really is system and drug-dependent components. And what we can do is investigate the impact of intrinsic and extrinsic factors on these system components that then obviously impact on the PK. And that is why we're interested in looking at PBPK modeling, because what we do is we separate out these components, build mechanistic models that then allow us to have a look at these intrinsic and extrinsic factors such as smoking, such as disease, alcohol-related issues, and so on and so forth. And hopefully this will become clear as we go through the examples that I'm going to show you today. So when we're actually doing model development, there are a number of models that we can actually use and that have been published in the literature. And this first one that I'm going to describe here is a minimal PBPK model. And this is generally used when we're actually focusing on drugs that have a low volume of distribution and aren't extensively distributed into tissues. And what we've got are three compartments. We've got the portal vein, we've got the liver, and we've got the systemic compartment. So if you take a drug orally, it goes into the GI tract, you've got first-order absorption, controlling it into the portal vein, and then it's carried to the liver where you get hepatic clearance, and then, of course, it circulates in the systemic compartment. And this is a relatively simple model that we can use to describe the disposition of a drug, and especially one where we've got a low volume of distribution. In this next example here, you can see that we've got a full PBPK model. And what we do here is divide the whole body up into these different compartments here, which are representative, the lung, adipose, bone, different organs within the body. We consider our arterial blood flow into these different organs and then venous blood flow as well. What we've done within this model is that within the liver and the gut, the brain and the kidney, where transporters play a big role, what we can do is actually incorporate a permeability limited model. And what that means is that we build in a more complex model with more compartments. So within this full PBPK model, within the organs, we break them down into additional compartments so that we can actually control or investigate the transport of drugs across these membranes here. So if we consider we've got the blood compartment here, we've got extracellular space here, and we've got the intracellular space here. And what we're looking at is this component here, which describes the active uptake or the transport of drugs across into the liver, and then they can be actively transported out into the bile. And this is the model that we would use if we wanted to have a look at transporter-mediated disposition for a specific drug. Now, when we're using this model, what we do is we have to define the volume terms of the different organs, but also we have to describe the partitioning of the drug into the different compartments. So if we consider this next slide here, what we're looking at here is the generation of these different tissue volumes here. But we have to have a relationship against the characteristics of the individuals, because what we don't want to do is just generate volumes randomly and independently. So as I mentioned, what we're interested in with the modeling is not only building the models themselves, but also incorporating variability into these models as well. So within the SimSip simulator, what we do is we use Monte Carlo simulation, and we generate the tissue volumes and other parameters required for PBPK modeling based on the demographics of the individuals and other covariates. So for example, if we think about tissue volumes here, what we would do is if we know the age, gender, weight, height of an individual, we can extrapolate this to a liver volume using equations such as these described below. Now, this is important because obviously if we have a look at these different organs here, what we've got are distributions with respect to the size of the lungs, size of the liver, size of the stomach, the GI tract and heart, and also the rest of the organs. And what we do is we sample from these distributions and assign these organ sizes to individuals, virtual individuals, within the SimSip simulator. But as I mentioned, it's very important to ensure that rather than just sampling randomly and generating these volumes, what we want to do is actually use a correlated Monte Carlo approach. So if you think back to these distributions here, what we're doing is we're assigning a volume based on the demographics of an individual rather than just sampling randomly and independently of any covariates. Because what we don't want is to end up with this situation here where we end up with tissue volumes for an individual that are completely wrong for that individual. And you can see here that in some cases the heart and lungs are too small, whereas the GI organs are far too big. So this is why we use a correlated Monte Carlo approach, so that what we can do is generate organs that are truly representative of that individual, and so on and so forth for a pediatric population as well. So this is exactly what we mean by a Monte Carlo approach versus a correlated Monte Carlo approach. And if I show you this next slide here, what we've got within SimSip is the relationship here. So we start off, if we're looking at generating a population, a brand new population, the first thing that we would do is have a look at the age-sex distribution of that population. We would have to know how the height correlates with the age, the weight correlates with the height. We would then use those two parameters to generate the body surface area that then would be used to extrapolate to all these different the organ sizes, as well as these different physiological parameters here. So this is exactly what we're looking for, because when we're developing a population, what we do is we have a look for real databases with real data in there that we can use to assign or generate the distributions, and we can take from those distributions and assign them to individuals based to the virtual individuals. But obviously what we want to do is try and look for these covariates so that we can assign appropriate volumes based on their body surface area or their demographics. And this is exactly what we do within SimSip. And of course, importantly, the different types of population that we'd be looking at, we'd be wanting to know what the impact is of ethnicity and disease on these different covariates here, so that we can build those into the model and then have a look at the impact ultimately on the system parameters and then the PK of the drug, exactly as I showed in one of the earlier slides. So what I'm going to do now is just mention briefly mechanistic absorption. I've talked about distribution kinetics and how we can represent that using a lumped minimal model. We can also use a full PBPK model if we're interested in incorporating transporters, or we have a drug that has a relatively high volume of distribution, or we want to know how that drug is partitioned into the different tissues. But what about the absorption side of things? Well, we have in SimSip the ADAM model, that is the Advanced Dissolution, Absorption, and Metabolism model. And what we do is we look at so we've got the GI tract split up into these different compartments here, including the colon and the stomach. And what we look at here is building the different physiology in here across, as we go across the GI tract, and we look at segregated blood flows as well. So we also consider the different regional PGP distribution and also CYP3A distribution as well. And we look at the amount of drug absorbed out of each compartment and also the metabolism within each compartment as well. So if we consider that we've got the solid dosage form, and then we want to actually consider the fine particles, release into fine particles, that is all incorporated within the ADAM model. We then have the dissolved drug, which we can have a look at the dissolution, consider the precipitation and supersaturation characteristics, but also consider degradation of the drug as well, as we know is quite common with some drugs. In the gut wall itself, we can consider the absorption, efflux and influx, and also metabolism. But as I said, importantly, what we've got built into the model is variability and also regional distribution with respect to the radius, the pH, permeability, enzymes, blood flows, and transporters. So within the PBPK models and also the absorption model, specifically the ADAM model, we've got permeability limited models that can account for transporters and also metabolism within the organs as well. But importantly, as I said, we do consider variability where appropriate, because at the end of the day, what we're interested in is looking at true variability across the population and not just what happens in average man. So what I'm going to do now is, this is just to show you then obviously what happens is the absorbed drug and the metabolized drug then gets incorporated in the portal vein, liver, and then taken through into the full PBPK model. What I'm going to do now is show you exactly what we mean by the top-down, bottom-up approach. And really, I mean, what we do mean by this is, when we talk about bottom-up, what we're talking about is extrapolating in vitro data. And then, of course, but using top-down data, what we can do is also use the clinical data to optimize the model and then apply this prospectively for simulations. So that is what we're talking about by top-down, bottom-up. So to start with, we can use a pure bottom-up approach. We're extrapolating the in vitro data, and if we can't recover the observed data, then what we can do is use that to actually optimize optimize the model. So importantly, and one of the things that we're always asked for is, and in fact, I think with respect to PBPK modeling, many people think that the number of parameters required to actually drive these models is quite considerable. So what I'm going to do here is just show you exactly what I mean by exactly what kind of data are required to actually drive a PBPK model. So if we want to predict a drug-drug interaction, and we're considering a victim drug here, I've already mentioned to you that when we're predicting out a volume, especially using a permeability limited model, then we calculate the system parameters, the volumes within the software, but the drug-dependent data that are required are physicochemical parameters as shown here. And these are actually used to calculate the partition coefficients, the partitioning of the drug into each of the different tissues. If we then consider the clearance component here, we need to have some idea of how the drug is metabolized with respect to human liver microsomes or hepatocytes or our CYP enzymes. But also, if we're wanting to look at a CYP-mediated drug-drug interaction, then we have to know the fraction metabolized by that CYP enzyme, and that has to be quantified precisely. We can assume first-order absorption and drive the absorption by a first-order rate constant, Ka, and assume an FA value based on CaCO2 permeability. But if we want to build in PGP and JMXKM and the solubility aspects, then we have to use a mechanistic model such as ADAM. So simply to represent the absorption side of things, we can assume first-order absorption or use something as sophisticated as the ADAM model if we want to have a look at formulation effects or incorporate PGP as well. If we want to build in the transporter kinetics of a drug and use the full permeability limited model, then the data that are required are JMax-KM data or intrinsic clearance data representative of efflux or uptake via a specific transporter. And also we need to have a look at how we can actually, at the relative contributions of the passive versus active. So those are the input data requirements. Now, that does look quite extensive, but in actual fact, if we look for repaglinide, and in this particular case here, you can see that these are the parameters that we're actually inputting in there to drive the model initially. So you can see we've got the physicochemical parameters, a plasma fraction unbound, we've got the blood-to-plasma ratio, and also we've got some idea how the drug is metabolized. So this is actually human liver microsomal data split into a 3A4 component and a 2C8 component, because we know that repaglinide is actually metabolized by both CYP3A4 and 2C8, and therefore what we have to do is make sure that we have these assigned accordingly, especially when we want to go on and do DDIs. And then, of course, we've got the CaCO2 permeability here. So just to show you how we would actually scale the data internally within the model and incorporate variability, we've got the data from the enzymes here, and that was taken from this publication. The next scaling parameter that we would apply would be CYP abundance, and this is the amount of P450 per milligram of microsomal protein. Now, what we're showing here are just average values. But of course, within SimCyP, we've actually got the mean and the CV values for distributions. And therefore, what we do within the software is we actually assign a different value for an individual from a distribution. And therefore, we end up with a range of CYP enzymes. The next parameter that we would apply would be the milligrams of microsomal protein per gram of liver. And we've got a relationship with age. So once we've generated the age sex of an individual, we would extrapolate an MPPGL value against age and then build that in. The last parameter to be added on is liver weight. Now, I'm showing you this because this is the way that we actually generate the FM values within SimCyP. We take the in vitro data, we scale it by the CYP abundance, and we multiply by the other parameters here to end up with a whole liver intrinsic clearance, which defines ultimately the FM value for the different CYP enzymes. In this next slide here, we know that repaglinide is an OATP1B1 substrate, and therefore it's important for us to build that into the kinetics of the drug. So once we've got the in vitro data, what we would do is then apply what we call a ref factor. And this is a relative expression factor in the hepatocytes relative to the in vitro system itself. And this is important because we have to make sure that the next point that what we're doing is correcting for the abundance values of the transporters in the system that we're measuring relative to what we're actually seeing in the liver. The next parameter that we would scale by would be number of hepatocytes per gram of liver, and we would have this relationship with the age. So if we know the age of an individual, we can extrapolate that to the appropriate HPGL and multiply throughout. And then finally, as in the previous slide, the last scaling factor would be the liver weight. So this is the process that we would actually go through. You take the in vitro data and scale it up to a whole organ clearance, and we would feed that into the model. The problem that we have is that for repaglinide, even though we know clinically, and there is certainly data available in the literature to show that the OATP1B1 genotype has an impact on the exposure, but yet when I went to have a look in the literature for the for JMAX-KM data, or indeed any hepatic uptake data for repaglinide, there weren't any available. So as I mentioned to you, what we can do is we can use, and this is actually just demonstrating a top-down, bottom-up approach here, where we're using clinical data and the in vitro data and fitting to actually get an unknown, to get the missing component. So we know that OATP1B1 is involved in the disposition of repaglinide, and indeed you can actually see significant differences in the exposure according to the genotype shown in this paper here. So even though there weren't any in vitro data available, as I said, clinically we know that it is important. So what I did was I fixed all of the input parameters that I'd shown you previously from the table. I imported this clinical data here, and then what I did was I optimized this value here. So I fitted for the OATP1B1 component. So I was using a full PBPK model, a permeability limited model for repaglinide. I imported all of the data, the in vitro data that I showed you from the previous slide, and then what I've done is I'm using this clinical data here to actually fit for an OATP1B1 intrinsic clearance value. And this is the value that I came up with. Now, when I actually did this, I didn't know what value I was going to come up with, but I was pleased to see at least that the value that we actually derived was similar to values that have been actually reported for OATP1B1 substrates in the literature, certainly in vitro. So at least that was the first step to showing that at least we didn't come up with a nonsensical value. And this is an approach that we've used. So at this point, we're able to recover the using the models, using the input data, and incorporating this OATP1B1 intrinsic clearance value or hepatic uptake value. We are able to recover observed data for repaglinide over a dose range, but that's with respect to single dose. Now, what the important thing to consider is if we actually want to go on and predict drug-drug interactions, we have to ensure that the relative contributions of CYP2C8, CYP3A4, and OATP1B1 are appropriate to clearance of repaglinide. And therefore, what we need to do is to predict drug-drug interactions with inhibitors of CYP2C8, CYP3A4, and OATP1B1. Now, the important thing is, before we can actually go on and do that, we have to ensure that we have models describing the kinetics of trimethoprim or any CYP2C8 inhibitor. In this particular case, we developed a model for trimethoprim because this is a specific CYP2C8 inhibitor. Because what we wanted to try and do is use inhibitors directed against a single enzyme or a single mechanism. So we developed a model for trimethoprim, clarithromycin against CYP3A4, and cyclosporin. Even though cyclosporin is an inhibitor of CYP3A4, it's a more potent inhibitor of OATP1B1. But before we could actually do any of that, as I said, we had to validate all of these models, again, independently against other substrates. So it is quite an involved process when we're actually doing the PBPK modeling. But this is just to show you that we've got the validated the validated inhibitors here with respect to recovery of observed data, but also the predicted DDIs as well. So in this next slide here, what I'm showing you is on this axis, we've got fold increase in AUC. We've got the inhibition that we're actually predicting from trimethoprim, clarithromycin, and cyclosporin. So these blocks here are showing the predicted data, and these are showing the observed data, because we have observed data available for all of these drug-drug interactions. So trimethoprim, we're predicting a value of about 1.4-fold. This is obviously on average. For clarithromycin, we're predicting a value of 1.3-fold, and the observed value is 1.4-fold. On average, for cyclosporin, we're predicting a value of about 2-fold, and the observed data is 2.4-fold. So really, with respect to the predicted mean fold increase in AUC, it's reasonably consistent with the actual observed data. What you can see here is that if we actually then go on and simulate out a complex DDI, including all of these inhibitors here, this is essentially what we're getting. We actually get a 6-fold, because what we're doing is knocking out all the 2C8, the 3A4, and also we've got the the transporter-mediated component there. But I think what is important as well is that what we predict, I mentioned that we're interested in predicting the range in a simulated population, and the maximum AUC ratio that we actually predict is about 11. Now, this approach that I've described here for repaglinide, where we're actually fitting for a transporter-mediated component, has been used by some companies. There were some papers published recently by Pfizer, Varma et al. 2012, where the drugs that they looked at were pravastatin, and also similarly they looked at repaglinide as well. And I think this is important because we know that there are issues with measuring transporter-mediated uptake and efflux in vitro. And therefore, this approach is being used more frequently at the moment until we can resolve those issues to actually fit for the transporter component. And therefore, it's important to actually validate the model. And as I said, I mean, this is something as well that has been discussed at the FDA and recognized that it is an approach to start with, and we do need further investigation. But really, what I'm trying to show you here is that what we have done is we have validated independently the drug-drug interactions between these inhibitors and also repaglinide, and therefore validated the relative contributions of CYP2C8, CYP3A4, and OATP1B1. And the reason why this is important is because if I go back to this slide here, what we're trying to do is have a look at the impact of intrinsic and extrinsic factors on PBPK models. So in this particular case, what we've done is we have developed a PBPK model for repaglinide. We've specified what the drug-dependent components are, and we've built in the system components. And we validated that model using healthy volunteer data and data that were available in the literature. Now what we can do is have a look at we can then go on and simulate in special populations and have a look to see what the impact of these intrinsic and extrinsic factors are on the system components, which then will obviously impact the PK. So in other words, we can then go on and simulate what happens in a renal impaired population, mild, moderate, severe, because we know that in a renally impaired population, we know that obviously we've got the renal clearance is impacted. We also know that we've got circulating uremic toxins that impact on the CYP enzyme activity and also transporter activity. We also have changes in the binding of a drug as well. For hepatic impairment, we've got child PU A, B, and C to consider, and we know that obviously because of the etiology of the disease, that the CYP enzymes are also impacted, and also the liver physiology as well. We can also we know that there are portal shunts that occur, and all of these we've actually got built into PBPK models so that we can assess the impact of hepatic impairment. With respect to pediatric population, obviously they're different to adults, and importantly, it's the ontogeny that we've got to build into the models here, and that is something that we do have within SimSip itself. Also, if we know what happens in an elderly population, we know that we get a change in the liver size, we get a change in the blood flows. All of this can be built in to the PBPK model into the system parameters, and therefore we can simulate out what impact it's going to have on the PK, and similarly for an obese population as well. So I think this is, so really, even though what we started with was a model to predict out DDIs, what I was doing there was trying to ensure that I got the relative contributions of the CYP enzymes and the transporters, and ultimately, now that we've validated that model, if we know how the population, the special populations, impact on those system parameters, we can actually then go and predict those out using the PBPK model that we've developed. The second example that I'm going to show you now is, as I mentioned, it's a real-life example. It's a drug that is in development at the moment. So I'm just going to show you a few slides here on a background for this drug B. So it's in development for hematological malignancies and inflammation. It's a CYP3A4 substrate. It's also a time-dependent inhibitor of CYP3A4, and it's a competitive inhibitor of both CYP3A4 and CYP2C8. So the first thing that I'm looking at when I see this data is that we need to obviously assess the potential for DDI with it as a CYP3A4 substrate, but on top of that, we have to build in the auto-inhibition of the CYP3A4 as well. And then obviously, because it's a competitive inhibitor of both CYP3A4 and CYP2C8, we'll probably want to assess the DDI potential of it as a victim, as a perpetrator against drugs such as midazolam and repaglinide, which is a CYP2C8 inhibitor, CYP2C8 substrate. So really, when we're developing a model for consultancy, the first thing is, what is the main aim of the project? Because I showed you some complex models at the beginning of the talk, so that we could either use a full PBPK model, we could use ADAM to actually fully characterize the formulation side of things. But at the end of the day, the more complex the models that you build in there, the more information and the more uncertainty potentially you could build into the model. So if we can get away with using a simpler model, then why not do that in the first instance? So this is why I'm asking, what is the main aim? Is it a victim or a perpetrator? Because as I indicated, if it's a victim drug, then we have to be able to know what the the contributions of the CYP enzymes of the transporter. We have to ensure that they are correct. We need to, what are the key components that we actually need to build into the model? What is the uncertainty? We need to identify the parameters with the most uncertainty. And as I mentioned, we have to develop either a full mechanistic PBPK model or a fit-for-purpose model. And that goes back to exactly the point that I made. Do we need a full PBPK model with permeability limited model built inside and ADAM, or can we get away with using a minimal PBPK model and assuming first-order absorption? And that is very dependent on the characteristics of the drug and the question that you're actually trying to answer. So the in vitro data that are available for drug B, there were metabolism data for human liver microsomes, hepatocytes, and recombinant CYP enzymes. Its FM value that was calculated ended up being in the range of about 70 to 80%. Fraction unbound was measured over a range of concentrations. We had the physicochemical parameters available, CACO2 data, and also inactivation data for CYP3A4. And as I mentioned, this is important because we have to build in the auto-inhibition of the drug, because it's a mechanism-based inhibitor of CYP3A4, but also a substrate of 3A4. So those were the in vitro data that we had to hand. What clinical data do we have that actually we can help us to actually build or validate the model? So this is a clinical study that was conducted. It's a 4-part study. There were the usual single ascending dose data, and there were 6 dose levels measured. Multiple ascending dose data, 4 dose levels measured. Also, food effects were looked at as well, and also a drug-drug interaction with ketoconazole. So this is the clinical data that I had a look at, because what I wanted to make sure is that I had this data available to help develop and possibly refine the model once I'd assessed how well the in vitro data were performing. So because I knew that it was a low volume of distribution drug, what I went for, and I wasn't building in transporter-mediated components into the model, I started with a minimal PBPK model. And what I was using to drive the hepatic clearance of the drug was the metabolic intrinsic clearance from human liver microsomes. We had CACO2 permeability that we were using to actually predict out to, and we were assuming first-order absorption. And also, we had physicochemical parameters available that allowed us to predict the partition coefficients and ultimately the volume of distribution. So we started off with a pure bottom-up approach where we're using all of the available in vitro data to extrapolate up to see how we actually recovered the data. Well, the first thing that I noticed was that the metabolic intrinsic clearance data, when we scaled that up to the an oral clearance value, we were under-predicting by about 30%. So what I did at that point in time was I used the oral clearance data and extrapolated back to a metabolic intrinsic clearance and incorporated that as an HLM input. So, and similarly, what we noticed was that using the physicochemical parameters, we had a predicted value of 1.51 liters per kilogram. But even though we didn't have an observed VSS value, we have had an observed VZ over F value of 0.61. So quite clearly that was telling us that our predicted volume was out. Therefore, what we did was we took all of the single-dose data that we had available for drug B at 4 different doses, and we incorporated this within SimSip. So this is the single-dose data taken from the single-dose clinical study. This is the structural model that we've had. I told you we were using the minimal PBPK model and assuming first-order absorption. So we had the structural model. We input the data that I've shown you for the from the table previously for drug B. But also on top of that, what we're building in is we're pulling in the clinical data and we are fitting. And what we're fitting for is this volume of distribution, because at the moment that is one of the parameters with the most uncertainty. And we ended up with a fitted volume of 0.18 liters per kilogram. So what I'm showing you here is that we started off from a pure in vitro point of view, extrapolated up, predicted a volume, clearance, and also FA and KA values, imported those into the model, compared them with observed single-dose data, and realized that the in vitro data were not able to recover the clinical data. We went back and evaluated the situation, realized that we were under-predicting the clearance and also over-predicting the volume. We used the clinical data and a fitting approach to come up with an optimal model that described the single-dose data. Now, at this point, what we wanted to do was obviously consider the multiple dosing of the drug. And when we're looking at the multiple dosing, what we have to build in there is the autoinhibition. So we have to consider a time-dependent or mechanism-based inhibition. And for mechanism-based inhibition, what we've got here is this is the enzyme, we've got synthesis of the enzyme, and we've got the turnover, we've got degradation of the enzyme. And normally in competitive inhibition, what happens is we get enzyme binding to substrate, release of the product itself, and then also release of the enzyme. And the same with competitive inhibition. We always maintain the same enzyme level. However, during mechanism-based inhibition, we get a metabolite that binds irreversibly to the enzyme, and therefore what we end up with is accelerated deactivation of the enzyme itself. So to model this in SimSip, what we do is we use this equation here. So we've got the rate of synthesis, we've got the rate of degradation, but also we've got this additional component here, which is the rate of inactivation. So this is the system parameter, and then this is the drug-dependent parameter. And what we're doing at each inhibitor concentration, we are calculating out what the K observed value is. So the KDEG value that we need, we reviewed the literature and found these values here. So this is actually the enzyme degradation rates, and you can see that the numbers are highly variable depending on the actual study that we look at. So this is the half-life here, this is CYP3A4, this is the number that we're interested in, and the half-life values range from about 36 up to about 94. So there is a huge amount of variability there. So before we can even go on and actually simulate the autoinhibition, we have to make a decision about the appropriate turnover number to actually use to model the autoinhibition. And what we did here was, this is from about 30 different published clinical DDIs involving time-dependent inhibitors of CYP3A4. We simulated them out in the software. So this is predicted AUC ratio versus observed AUC ratio. And these are the two extreme values that we used here. And you can see that clearly this KDEG value here, the one with the open circles, gives much better predictions than this value here. And therefore, this is the value that we've applied in further simulations of drug B. So this is what the simulations look like now with observed data overlaid. So during the multiple dose study, what happened is that, so this was actually the dose X that was given on day 1 and then given twice daily from days 2 through to 13. And so this is what the simulated data looks like for the full dosing profile. This is what it looks like on a semi-log scale on the last day of dosing, which is obviously what we're trying to recover. So previously we've shown that we were able to recover the single dose data. What we're trying to show here is that we can recover the multiple dose data. And what we've done is simulated out different trials of individuals based on the number of subjects that were actually used in the clinical study and also the characteristics. And you can see certainly at this particular dose here, we were able to recover the observed data reasonably well. Again, when the dose was increased by 5-fold, a similar plot, this is the multiple dosing data, and this is the actual semi-log plot shown on the last day. And you can see certainly that with respect to the trials, we are able to capture the observed data reasonably well. And again, we use the characteristics of the individuals in the actual study. So at this point, we're able to demonstrate, I haven't shown you the other doses, but we did a similar exercise for the other 2 doses, and again, we were able to recover the multiple dosing data. So the next step is what actually happens now with the DDI study, because I mentioned in the clinical data that they had available, they performed a clinical study with ketoconazole. Now, in what they'd actually done for that clinical study was they'd given 5 days of dosing of ketoconazole, 200 milligrams BID, and then they'd given a single oral dose of their drug on the 4th day of 5 days of dosing. And what I'm showing you here in this plot is this is the predicted AUC ratio. These open circles are the simulated trials, and this is the actual observed study here. So what I'm showing you here is the variability, because as I mentioned to you, what we're interested in is not only predicting the mean or the median, but also predicting out the what we may see. So what we do is we simulate all of these different individuals, randomly assign them to trials of 16 individuals, which is the number that was actually used in the clinical study. And then, of course, what we do is then show you that how the median value and the range actually compares to the observed data. So the model that we developed using single dose data, we're able to recover the multiple dose data, and now what we're showing you is that we're able to recover the study with ketoconazole. But I had a concern about the design of this study because this is single dose data, and we know that during multiple dosing of drug B, we're going to get autoinhibition occurring, which is going to lead to reduction in CYP3A, and therefore you may get a different result when you perform a multiple dosing study versus single dosing study. So let me just show you what I mean by this in this next slide. If we consider, on this graph here, we've got the amount of active enzyme. This is the control situation. So this is what is happening when you're just giving drug B itself. It's auto-inhibiting. It's knocking out its own enzyme. Then when you give ketoconazole, this is what's happening to the enzyme. So this is what would happen if you gave single dose of a drug, a single dose of their drug with ketoconazole. You still got that amount of CYP3A4 enzyme, whereas during multiple dosing, so if you multiple dose their drug to steady state and then give the ketoconazole, you can see that because you've actually got autoinhibition of 3A4, the FM is lower, and therefore the potential for drug-drug interaction with ketoconazole is lower. So if we then go on and have a look at the dose-dependent effect, you can see that at X mg BID, the result was 4.1. When we increase that dose fivefold, we're getting a decreased interaction. And ultimately, at 25 times that dose, then we actually decrease the predicted interaction is 2.4-fold. And let me just explain that to you on the next slide. What is happening is that we've got, at this particular dose here, you're knocking out this amount of enzyme. This is just considering the drug itself. However, when you go to a much higher dose, what is happening is in this particular case, you can see that you're getting much more of the enzyme knocked out. And therefore, the FM at this particular point, when you're giving the ketoconazole, is much lower than in this case. And that is why you're getting a decrease in the drug-drug interaction with ketoconazole. So even though they'd already conducted a clinical study, by developing this model, we were able to inform them what would happen with other study designs. Importantly, another point that the client was interested in is if we've got data with a strong inhibitor, what happens with a moderate inhibitor? And can this be supported by simulation? So these are the results that we've actually got with ketoconazole. So we've actually got a model developed for fluconazole. And what we were able to do is show that as you go from 1x to 25x, again, we see this susceptibility to drug-drug interaction decreasing, but not as strong as in the case of ketoconazole, because what we're looking at here is a moderate inhibitor versus a strong inhibitor. So this is just to show you an example here of how we've developed a model, how we've used clinical data to refine the model, but then gone and applied the model prospectively to inform on clinical, on the study design for prospective clinical studies, including investigation of a moderate inhibitor. So what I'm going to finish off with now is just a quick look at the regulatory perspective here. So you'll be pleased to hear that I've just got a few slides here. But I think it's important to show you really where we are with PBPK modeling in the regulatory view. So these were two slides that were presented by Xumei Huang from the FDA at the APS meeting last week. These data are publicly available. This is the number of, so this is the regulatory submissions that were done with PBPK data. Essentially, this is the breakdown here. So most of the submissions that were done with PBPK data were regarding DDI, but importantly, there were an increasing number with pediatrics, hepatic impairment, so this goes back to the special populations that I was talking about, DDI and pharmacokinetics on its own, and also absorption side of things. So this is just to show you the area of the PBPK modeling. In the next slide, what I want to show you is the number of submissions that have been done with PBPK. So this is 2008 and this is 2012. So this represents here a part of an IND submission that were actually done by the sponsor, and this is an NDA submission. So in 2012, there were 4 IND submissions and 4 NDA submissions that were actually done by the sponsor. But I think what is really important here is what they're showing you is that if submissions weren't done, including PBPK modeling, what is happening now is that the reviewers at the FDA are developing models, are developing PBPK models to actually explain some of the data themselves. And this certainly, in my view, lends further to support that the FDA is endorsing, really, the application of PBPK modeling during drug development. And certainly, if you aren't looking at it, they will be looking at it, certainly to answer some of the questions. And I think one of the other issues as well that I haven't really talked about, but certainly is important to consider, especially with respect to PBPK modeling, is metabolite kinetics, and I know that that's certainly something that they've been looking at. Okay, so I'd like to, so that's where I'm going to finish off, and what I'd like to do is acknowledge my colleagues at SimSip and thank them for their contributions.